Good morning, everyone. Thanks so much for joining us. We'll just wait a minute or so to let people start filtering into the room, and then we'll get started. Okay, so I think the attendees have started to even off a little bit now, but I'm sure we'll have some more people joining us. Well, thank you very much for coming along today for this webinar, where we're really going to focus on the power of data in environmental sciences. The purpose of this webinar is really to encourage collaborative research across data and life sciences. And in true fashion, we have put in a collaborative effort to run this webinar today. So it is a partnership between the Data Lab, which is Scotland's Innovation Centre for Data and AI, and SULSA, which is Scotland's research pool for life sciences. So to give a little bit more of an introduction to that and to myself, I will start by saying that I am Ali Hughes. I will be your chair for this morning's webinar. I am currently the Strategic Partnership Manager for SULSA, which is the life sciences research pool in Scotland. So we are a strategic partnership of 13 higher education institutes in Scotland. And we act as a responsive platform to leverage funding, create opportunities, and to lobby on behalf of the life sciences research community here in Scotland. And we've been really enjoying this partnership with the Data Lab, and in particular with the Data Lab community, which is an online community of over 6,000 data and AI professionals, students, and enthusiasts, so going across sectors as well. And the community runs regular events, so webinars like the one today, and then you'll be able to find like-minded peers, be able to ask questions, kind of interact and start building your network within the data community. So today's session, as I said, it's really going to be focused. We're really looking to inspire and encourage you to look at how data sciences and life sciences can come together to create some really impactful research. And we've got some great speakers for you this morning. I keep going to say this afternoon. I think the bank holiday has really messed up my timing. So yeah, we are going to hear from leading researchers on the use of genomics, ecological modeling, and environmental informatics to drive new insights into the natural world. And you'll see how kind of data sciences and life sciences really complement one another in this work. So just a couple of little housekeeping bits. This is a webinar. It is being recorded. We will encourage you to use the Q&A box. So hopefully on the bottom of your screen, you can see a Q&A box. Please pop any of your questions into there. We'll have time after each of the speakers to ask some questions, and then we can have a more general maybe Q&A or panel discussion for the last few minutes of the webinar as well. So yeah, please do. We will keep an eye on the chat box as well, but please do direct any questions into the Q&A so that I don't miss them with all of the chat. So we'll get straight into it anyways and introduce our first set of speakers. So that's Dr. Ivan Puchernik and Dr. Bruna Santana. Dr. Ivan Puchernik is a career track fellow with the Roslin Institute and is interested in the development of novel breeding methodologies and tools. And Dr. Bruna Santana is doing her KTP associate with Aquanzo and the Roslin Institute and is an animal scientist by training, specializing in quantitative genetics and breeding. So I will hand over to you to give your talk. Thanks very much.
Thank you for the introduction, Alison. I'm going to share my screen now, and then I hope everybody can see our screen.
Yep.
Perfect. So thank you for introduction again. So first, because this event is kind of to motivate collaboration between the data and AI scientists and experts in the field with the academics, I want to introduce our two teams. So basically here at the Roslin Institute, we have a Highlander lab led by Professor Goriantz and Potrinich lab led by myself. and two of our labs are working quite a lot with industry collaborators on implementing quantitative genomic models for selective data-driven breeding. And one of these projects is innovative UK KTP partnership between University of Edinburgh and Aquanzo, and this is where Dr. Santana will present few of the projects there. So we are located at the outskirts of Edinburgh, at the Easterbush campus, at the Roslin Institute, and if you don't know for anything else, Dolly sheep, so first cloned mammal, was developed here, and we like to say that we are pioneering animal biosciences. One of the examples that we have from last few years, that we are proud that for every £1 of public funds we receive, we generate 3.4 pounds within Scotland and 13 pounds across the UK. So what do we do? So basically we do data-driven selective breeding. So we employ various quantitative genetics models to select the best animals that are going to be the parents for the future generations, with the goal of improving that population for certain trait, usually connected to health of the animal or production of the animal. And historically, what we did, we wanted to understand what animals have the best genotype, and because we could not know that at the time, we used whatever was measurable and observable, and I'm going to call that phenotype of the animal or individual. For example, here you see a bucket of milk as an example. And then what do we do with those data sets that we collected? We would basically input them in our black box machinery, our predictive algorithms that would allow us to rank the individuals and select the best ones. And when we select the best individuals, whatever the best means, over the course of time, over the generations, we would see improvement in the trait, or in this case, we would get 2 buckets of milk instead of 1 bucket of milk. And to show you a real life example, this is from UK report for the UK dairy cattle, and please look at the blue line, where you can see clearly the increase in the milk production over the years, and about 50% of this increase can be accounted for genomic data-driven selection. So over the time, we went from this black box machinery that we use for the prediction based on just these observational phenotypes. We now know how to dig into the genome of the animal, genome of individuals, and we also have multi-omic approach today where we have many more data points. And this is where AI really is great, because we can use machine learning and deep learning techniques to disentangle this. But what we wanted to talk today is really computer vision, because this is where we have the biggest impact from the AI and machine learning, because some of the traits, some of these phenotypes that we want to record are very expensive and hard to measure. So imagine if we have cameras and sensors on farms, we can track behavior of the animals, we can see lameness of the animals. For example, we can see how animals eat, etc. And this is really where we think that biggest opportunities lay in our field. And this is quite old idea. So this is a paper from 1986 where one of the quite prominent researchers in the field of animal breeding and genetics applied at the time very advanced TV cameras and TV monitors to try to record images of the cows and try to use the computer algorithm to predict carcass composition. So the idea is quite old, but now we really have the technical solutions to implement these things. I'm going to show first example, which is the dairy breeding project we have with our commercial partner Geno. Geno is a dairy cattle breeding company from Norway, and what the research question was, there are 2 very expensive phenotypes to record. One is the feed intake and the other one is methane emission. And there was a question, what kind of progress would they have after 10 years of increasing the number of phenotype records or data points of these expensive to measure phenotypes. So what we've done here, we use our in-house software called AlphaSimR to simulate to do a stochastic simulation, and really we did digital twin of Genomes breeding program, and we tried to see how increasing the number of phenotypic observations and data points would increase the progress in the traits. So for example, in this slide, I'm showing you the impact of current situation where we had 1000 phenotypes. The increase of improvement in this expensive trait is about 3 points. If we double the number of data points and recording of the expensive trait, we will see certain increase, and if we triple it, we will basically double the impact and progress in selection of this trait. So really, this is an exemplar project where AI and computer vision could be used, and really to solve this expensive phenotyping, where we clearly see that more data points are needed to make a progress. And now I would like to hand over to Dr. Santana to talk about selective breeding project we have in Artemia.
Thank you, Eva. So I'm gonna be talking more specifically about this project that I am working. So it is a partnership between the company Aquanzo and the Rosley Institute with the support of Innovate UK. So the goal here is to develop and implement a breeding program for Artemia, which is a little crustacean, also known as brine shrimp or sea monkeys. And computer vision and AI would be in the context of high throughput phenotyping. So just to some context, I'm going to mention some aspects important in terms of the breeding in the breeding scenario. So to genetically improve the populations, the strategy would be selecting individuals or families to obtain a better production, reproduction, and fitness related traits. So improving these traits in the population with time. So here I have this equation. Eva mentioned phenotype, genotype, and environment. So in this equation, we see that phenotype is an important part of the scenario. And for Artemia, for example, what we'd be interested in length or size of each individual, the number of shrimp, the speed in which they are swimming, and other characteristics. So phenotyping in this context can be challenging for the large number of shrimp, also for the short generational interval or short reproduction cycle, and also because they are not static, so the movement, so this can limit the feasibility of phenotyping. Just moving on. So the use of computer vision and AI in this scenario would enable the precise recording of phenotypic traits, and what we want is data accuracy and speed the data analysis to support decision making. And also this would open the way to improve to scale the program. So for scalability of the program, that would include in the future automatic tank sampler and data platform to for the analysis. Yeah, moving on. So what we want is to optimize what we have and produce new developments. So far, some work has been done with computer vision libraries that are open, and also deep learning models pre-trained that are available. but we also envision training new deep learning models, for example, for assigning sex, maturity status, and other anatomical features, healthy status. And the goal here would to be like understanding what it would be the conditions for filming and key parameters to apply this application in distinct scenarios and maintaining like high accuracy of the phenotypic information precision, and also in long term we would have a large volume of data, so optimizing the processes, the algorithms, and would be important. So yeah, thank you for bearing with us. Just a quick acknowledgement to those that are supporting us, especially UK Research Innovation and this knowledge transfer partnership. Thank you.
Thank you so much, Ivan and Bruna, for that. That was a really interesting talk and a really great one to get us started. If any of the audience have any questions, please do pop them into the Q&A. I guess one question that you've kind of mentioned that one of your future objectives is to optimize that data analysis. Could you maybe give a little bit more information about what you're hoping to do with that and where potential collaboration might be useful in doing that?
Yeah, we are looking for collaboration for people that have more experience in the development of applications and processing this type of data. But basically, we would have a few days to process a large volume of data. So like a fast process and efficient process would be something that we are looking for. And so far, we have an application developed, but it's only working for like a specific scenario. So often when we have new videos, we have really to take time to check several parameters and change parameters. So we wouldn't have time for that. So yeah, so that's where we are looking to improve.
Great, thanks so much. We do have a question here. So are you using standard or readily available methods for things like image recognition, or are you actively developing your own?
So the company was using OpenCV so far, which is available, and also starting to train deep learning models for object identification. So yeah, so we have something that we are using that is already available, but also try to develop new things. Yeah.
Brilliant, thank you. And do you currently have any funding available for collaboration? I think that's
Yeah,
already.
I think I'll take this one. So I think this kind of pops up almost always. So we have certain internal pump priming small grants for kind of proof of concept studies, as well as here at the Eastern Bush campus we have Campus Innovation Award, which our business development team is working on. So these are some options that we have, usually small scale grants to really show the proof of concept of the work. But we don't have any major open position at this time.
Yeah, and maybe just to follow on from that as well, that we are going to be at the end of the webinar talking a little bit about the Innovation Seed Fund, which is going to come at the start of May. So that is specifically for industry academia collaboration, but is looking at bringing data and life sciences together. So I'll give a little bit more information about that at the end, but is also another potential for that kind of pump prime money to get a collaboration off the ground. So yeah, any other questions that people want to pop in now before we move on to our next speaker? I had one that was just so my background is chemistry, and then I've done a little bit of microbiology. So eukaryotes are very much outside of my wheelhouse. But I was wondering in terms of if you have multiple genetic factors that affect your phenotype, do you have like good modeling around like kind of a design of experiments type model that you can use to sort of play with those? Or is it mostly experimental work that you're doing at the minute to get those phenotypes or to understand what affects those phenotypes?
Is that for me? Yeah, so right now we're just starting developing this program, and the goal is to collect phenotypes and environmental variables and start modeling, like statistical modeling, to understand what is really impacting the phenotypes. But yeah, so we don't have a lot in literature, but we are, we'll test a few different models and see like what would work best for the data that we have available. Maybe try to understand like potential additional data that we would need to collect for better like results.
This is a typical example when a new species is entering this kind of selective breeding, while on the other hand we have cattle, pigs, chickens, kind of classical livestock examples that are bred for many years, where we have quite good data records already available. But the data in those species is not from experimental, it is observable, right? We cannot control everything we have.
Cool. Thank you so much for that. So yeah, thanks again to Ivan and Bruna for that talk. We will have other opportunities. Oh, sorry, we've gotten one more in here before we move on. If you can talk a little bit about the ethical considerations that you need to think about when working with selective breeding. I guess it's an area that yeah, not a lot of people would be familiar in. So always interesting to hear about some of the considerations that are important in terms of ethics.
So yeah, definitely. Just to say, selective breeding is something that humans are doing for many hundreds and thousands of years. The moment that we domesticated animals, we basically started with a selective breeding. In this context, the methods that we apply in most of the livestock species currently are standard ones. So either it's natural mating, eventually artificial insemination. So we are not talking here about any novel methods like genome editing or things like that. So yeah, it's kind of very standard practices that we use that are done for centuries basically.
Thanks so much. Yeah, I think it's an interesting or an important distinction as well between the kind of selective breeding and then when you start getting into the genetic modification world of things. So yeah, so thank you very much for that talk and for getting our webinar started. We're now going to move on to our next speaker. We will have time, so please keep putting questions in the Q&A box. We will have time at the end to chat a little bit more and ask some more questions. But for now, I will introduce Tanya Riley, a postgraduate marine scientist student at UHI Shetland, and Dr. Rachel Shucksmith, an associate professor of marine management and governance. Their focus is really on enhancing our understanding of marine habitats and in a particular interest in the Shetland benthic communities and habitats with blue carbon potential. So I will hand over to Tanya and Rachel to bring you through their talk.
Hello, hopefully you should be able to hear me. I don't know.
Yeah,
Having some technical difficulties that we didn't have before.
No, just typical. Yeah, we can see your screen okay now.
Perfect.
Lovely, thanks.
Lovely. So, thank you for the introduction, Alison. So it's great to be invited to speak about the local data that we have here at UHI Shetland and how we use that to support marine planning and restoration in Shetland. So As has already been said, my name's Tanya Riley. I'm a marine scientist within the Marine Spatial Planning team at UHI Shetland, and I have experience and work within marine restoration in Shetland, benthic habitat mapping, different types of modelling and data analysis, as well as stakeholder engagement. And Rachel?
I'm Rachel Smith. I'm an Associate Professor of Marine Management and Governance here at UHI Shetland, and I lead the Marine Planning team where we do a range of interdisciplinary marine science projects, taking knowledge and data from a range of stakeholders to inform policy and decision making.
Fabulous. So to begin with, I thought we'd start with the project that we've just finished, which is looking at marine restoration in Shetland and kind of what could that look like, because marine restoration is still quite new. It's still a lot of unknowns. So we have just finished a project looking at what do local people want marine restoration in Shetland to look like. So the first phase, we had 160 participants from all across Shetland. That included marine industry, government agencies, environmental NGOs, community councils and local residents. And we kind of asked them what were the challenges and opportunities that they saw for Shetland specifically moving forward. And the main thing that was quite striking was that across all of these 160 participants in this first stage of engagement, there was kind of unanimous support for that they wanted baseline data sets, they wanted local data sets, they wanted long-term monitoring after the data sets to kind of keep informing what was happening in the marine environment in Shetland. It's kind of really deeply embedded, just kind of in everyone's way of life. You can't go anywhere in Shetland without seeing the sea. So it's a big part of everyone's life, as well as tourism and livelihoods. So it was really nice to see the community also saying this. So in phase 2, we kind of then went, okay, this is what people have said. How much do the wider population of Shetland agree with this? Because these were individual people being spoken to. And so the second round, we had 271 participants kind of say how much they agreed or disagreed with challenges and opportunities that had been identified in phase 1. And what we found was that they really wanted robust and long-term local baseline and monitoring data, and 87% of people agreed with this. And I think that was the main thing I kind of wanted to share with everyone as data scientists, is there was a lot of support for data moving forward and for things to be supported by local data. And therefore we created a guiding principle for marine restoration that recognised this as well. And I think this has also been shown in research as well. We did a little research project last year that kind of showed the importance of the spatial resolution of data when you're doing modelling. And so the sea, we haven't mapped all of the sea. We can't see everything in the sea, and it's highly dynamic and changeable compared to the land. So we have to use a lot of models to predict either where things are, how things could have changed, what future potential is, especially in the space of marine restoration. And that all depends on the baseline resolution of data that you have. So we were comparing, here we have 50 metre, 100 metre, 200 metre and 500 metre resolution. And you can kind of see that it becomes very blocky by the time you get to 500 metres. Like, you've lost all of these intricacies that you see at 50 metres. And therefore, when we ran it on a predictive model, so this was a machine learning model called Maxent, and it looks at the suitability of one area compared to another area for a habitat or species based on the environmental data. We see that the predictions change quite a lot with the resolution, and you get to say there's not much difference between 50 metre resolution and 100 metre resolution, but by the time you get to 200 metre resolution, this habitat could live everywhere in this area, and we know that that's not true. And at 500 metre resolutions, we actually have areas where it does occur, just simply disappearing. So the science is telling us that data is important. The public and community are saying that data is important. And I think that that's really well grounded in Shetland. In terms of marine biological surveys, we have a lot of annual, seasonal, monthly, and sometimes almost daily biological surveys that happen, not just with UHI Shetland, but we have SOTIAG, which was set up by the Sullombo Oil Terminal, the Shetland Amenity Trust. We've got whale and dolphin conservation that do shore watches looking for cetaceans. That's the most frequent one that could be done almost daily. RSPB looking for birds, Nature Scots, SMU looking at seals, and this is not exhaustive. Like, there is so many more surveys that are happening all the time in Shetland, as well as looking at like invasive non-native species monitoring, for which UHI Shetland established one of the only long-term marine non-native species monitoring programs in the UK, and that's still going to this day, to make sure that we have this high resolution data so that we can respond very quickly if anything was to invade appear in Shetland, or that we can start predicting trends of invasive species based on this fast responsive monitoring program. Another example of this in marine restoration is seagrass restoration, which you've probably all heard of at some point about seagrass and how important it is. So we've just completed the first stage of a project where we use local data to look at the historical extent of seagrass beds that used to be widespread throughout Shetland, and this was using archival records as well as local knowledge and asking community members. We then went to these places and used drones to survey to try and see if we could see the seagrass beds, because they're only in 5 meters deep of water, and the water in Shetland is really, really clear. And then when we found the bed, we then swam around it to map its extent. We were able to identify, sorry, that should say 15 beds, not X beds, 15 beds across 2 sites is all that's left in Shetland at the moment. And this data forms the base of us for looking forward in time to the potential for replanting seagrass. But this is all underpinned by looking at habitat suitability, preferences and stuff like that to inform restoration, as well as monitoring it annually. So we have ongoing drone surveys where we are aiming to track the annual growth of the seagrass bed, and its natural dieback every winter, because obviously it is an underwater plant. It does grow and then die back, which AI might be the answer for, but we haven't quite got there just yet, because this is all very, very new. So that might be something to explore later in the discussions. But that's kind of where we are at the moment in terms of marine restoration. And I'll now hand over to Rachel, that's going to tell you a bit more about all of our other data that we have in Shetland as well.
Thank you, Tanya. So much of our data in Shetland feeds into the Shetland Islands Regional Marine Plan, which is a statutory planning process that we are delivering on behalf of Scottish ministers. And the Shetland Marine Plan has been going since 2006, but the first 18 months of that process, and then continuously from 2006 to now, has been the collection and collation of data to inform marine management. So this has been very wide-ranging, including environmental and biological data, such as protected areas, marine mammals, seabirds, etc., geological data, oceanographic data, and information on infrastructure and marine use. One of the key findings of this data process has firstly been that without detailed data, you can't make detailed policy. So data is fundamentally important in developing policy that can guide marine decision making. Also, that many national data sets are at a low resolution and contained quite a number of inaccuracies, and often didn't include data that was relevant at a local level. So, for example, inshore fisheries was missing, recreation was missing, historical marine biological records were often inaccurately recorded. And over the last decade, we've been trying to identify and fill these data gaps to ensure decision making is made on the best available and accurate data. And we've made a roadmap going forward to ensure that these gaps are filled so that we can manage the marine environment in a sustainable way. One of the key findings within this, though, it has been that the need for local scrutiny of the data sets, and also to ensure that it's collected at resolution that allows national policies to be delivered at a local level that's meaningful. We back in 2014, when we wrote a paper detailing some of the steps and processes within this data collection process to inform regional marine planning. So if anyone's interested, this is the title of the publication.
Yeah. So looking forward in terms of what we have, Rachel's done a great job of identifying that we've got a roadmap to fill these data gaps, and these are the challenges that we face in terms of ecological issues moving forward, and in the marine especially, that we often have quite small or very, very patchy data sets. Some of them are historically dependent as well, which could have inaccuracies. Sometimes there's a massive issue in integrating diverse data types. So you might have satellite data, community input, in situ data, and it's trying to find a way that you can integrate all these different data types into one place. Data gaps in remote areas are really difficult as well, or if you want something really specific for an ecological process, and just the need for standardization across multiple data sources as well, to make sure that when you're bringing it all together, it's all in a standardized format. So moving forward, any collaboration in terms of enhancing our habitat suitability models or other modeling and predictions, especially looking at tools for forecasting, prevent potential environmental change, but also back forecasting as to what has changed in the marine environment, as well as any ways that we can look at integrating all these different data types together and showcasing that. So thank you very much for listening, everyone. Yeah, we take questions.
Thanks so much, Tanya and Rachel, for that. That was a really interesting talk, and very, very different to our first talk as well, while still sticking on the theme. So thank you very much for that. We have a few questions that have come in. One of them being, do you have information on the profile of the people that responded to your research questions? It's really interesting to see the general public is interested in the data. And I guess if I could tag on to that a little bit in maybe how you engaged with the variety of stakeholders that you had involved in those surveys.
Yeah, so in terms of the demographics, we didn't take any demographic information apart from obviously face-to-face knowing who people were. We took what type of marine user they were, so were they working in the marine industry, were they a fisher, were they this, what type of marine use did they, how did they use the marine environment, if that makes sense. And we found that most of the people that responded, on average, they engaged with the marine environment in at least 2 activities. So whether this was recreationally or for their job, they utilized the marine environment in more than 2 ways. We had a very diverse range of engagement. We had an online survey as the kind of final catchment to catch everyone. We had targeted discussions with marine industry, in fact, the government agencies and NGOs and stuff like that. And then we had lots of community drop-ins. So we went on a little road tour of Shetland, which was great considering I just moved here. I got to see all of the islands, where we were at community halls, and we'd advertised that and people came and spoke to us. We had evening talks. We had stalls that were just left up at the library so that people could engage with it without me being there to scare them away. So it was quite an intensive process to try and get as many people as possible to engage.
Yeah, absolutely. Yeah, fair play for bringing together so many people and getting such a wide variety of stakeholders to engage in it. So another one then that's looking much more at the kind of technical and data side of it is Are you using Earth observation data and merging that with your drone ROV with camera, your sensor data? And I guess maybe a bit of a follow-on question to that is that have you spoken to any of the kind of geospatial organizations in Scotland who might be able to help with some of that? An example there was AGI, Location Data Scotland, Map Impact, or Geovation Scotland. So yeah, just to chat a little bit more about that data.
Yeah, so I haven't heard of that organisation specifically, so I'll definitely look into that afterwards. What we have a process of doing is where we have data, we always try and overlay the local data on top of it so that we have a high resolution where possible, if that makes sense. So like in terms of bathymetry, we've got data from UKC sediment types, and then we've got the EU one below that that's even at a coarser resolution, and then we've got like our local surveys that we put on top of it to try and make sure that where we have the higher resolution data, we are able to, and we have like data sharing agreements where possible to try and bring that together. But any advice as to other data sets that we might not know of, or if other people have data sets, is definitely, we're like really open to finding high resolution data sets.
Yeah, absolutely. There's quite a few bits that are coming in here about the data side of things. So I definitely think there's some people in the audience that might be able to help you with some of this. Because another question related to that was around the methodology or the machine learning model that you use to identify the seagrasses from the drone footage. And that you kind of noted that there are different organizations that collect data around Shetland. So, you know, are there good kind of data sharing practices between you? You did mention that standardizing data is a difficulty, but have you been able to get any kind of standardization agreement between some key stakeholders to make that a little bit easier? And then are there particular data collection methods that you intend to use to fill some of the gaps that you have in the local data?
Did you want to do the local data one?
Yeah, I think the data we incorporate is so diverse, there isn't just one method we'll use. So we try to incorporate both local knowledge and data, and the methods we've used range from like drones, acoustics, public observations, fishermen doing participatory mapping, or using AIS or VMS. So it's a huge diversity of data that we collect together. And we try to adhere to the same data standards, though, within all data types, ensuring that the data is accurate and checked. But there isn't just one method we utilise to collect or to collate it.
And maybe just to give an idea of how much work is behind that, someone has asked how much time do you spend trying to bring together the data and pre-process the data versus actually, you know, kind of getting analysis and getting information back out of that data to support your research, if you can give a bit of an idea of the input in and the output.
I mean, I guess that's difficult to quantify because we've been doing, like from our regional marine plan, we've been doing it since 2006. So a lot of that process has been undertaken over a long period of time. And then, for instance, we had a checking process for the benthic data, which particularly related to protected habitats of horse mussel and mull, and that work stream's been going on for over a decade now as well. And that's required boat work to check the historical records and putting it in a format that's suitable. But new data that's being collected, for instance, by the government agency. So we looked at historical data that was collected 20, 30, 40 years ago, and that tended to be quite low spatial accuracy, whereas new data coming in is already compliant to MEDIN standards. So there's far less work with new data than there is with the historical data.
And everyone's very open for discussions and stuff. Like we just had a really massive marine crazy weekend with lots of different organisations coming up from down south, like Whale and Dolphin Conservation, the Scottish Marine Animal Stranding Schemes, RSPB. And at certain points it was like, right, what can we do to help each other? We're collecting this data, is it of use to you? What extra things could we put in place that would make it useful for you? So we've got lots of really good open channels for that discussion to make sure that the data is as useful as possible.
We do have formal data sharing agreements though with key organisations, particularly where it might contain personal data. And within that context, I guess that's an important part, is ensuring that the data ownership stays also with the data owner. So for fisheries, for instance, we always check with them how the data is going to be presented and how it's going to be used, because that's really important for them before they are willing to share data with us. So the relationships we've established have been over long periods of time, which has allowed us to access such a range of data sources.
Yeah, that's brilliant. Thanks so much for going into some more detail about that. And I guess since you have this sort of data that's over quite a decent period of time now, have you been able to observe any changes that you think might be directly linked to climate change or the result of climate change from that?
No, not in the times, not directly in the time series time that we have. Obviously, there are, I guess that can be picked apart from things like the bird data, for instance, although other organisations like the RSPB, for instance, lead on that. So not within our analysis so much, but within some of the data sets, yes, there are changes that could be attributed to climate change.
Yeah, that's fair. I'll maybe ask one more question before I bring our other speakers back in, because I think a couple of the other questions that are here might be of interest for them as well. But one was saying that it sounds a bit like you're building an Internet of Things sensors network. And how are you considering the kind of maintenance access to the sensors once you have those placed?
We have a number of projects running at the moment. For instance, we have Blue Connect, which is a Horizon Europe project, and that's looking at how we can use innovation and citizen science to collect data. So for most of our projects, we apply for and gain continuous funding to ensure that data can be collected and continue to be collected over a period of time.
Brilliant. Thanks so much. Sorry, just one more has come in here that might be of interest for yourselves. So this is someone who is working with natural and cultural data, so historic and contemporary, for terrestrial site restoration. It's great to be able to see your approach to modeling work. You may already know that under the new AHRC Riches programme, the UHI Institute of Archaeology will develop a UK Centre of Excellence for Geoarchaeology, so paleoecology and paleo-environmental monitoring. And so basically, Claire Boardman is wondering if there's any kind of productive touch points, with that data or between that work and your own work that might be useful going forward.
Yeah, we could certainly look into that, because the historical data does form part of our evidence base within the marine plan. So yeah, that would be interesting to explore.
Perfect. Claire, if you're involved in that project and would be interested in getting linked up, if you aren't already kind of in the one UHI network, very happy to make that connection for you via email if that would be useful. So I might just, for the last few minutes, we'll invite Ivan and Bruna back on camera with us as well. Just to have a little bit more there's a few more questions here that I think there might be some interesting inputs from both of you around a little bit more around that data, some of the machine learning and modeling that you can use where there might be some overlap between yourselves. So one question was, do you work with deep learning for non-image data? And if you have any tips on that. So this was targeted at Ivan and Bruna, but Tanya and Rachel might also have some insight into that. And if you have any tips for Natalia on that, would be great. I'll maybe hand over to Ivan and Bruna first.
Yeah.
Yeah, I haven't worked with non-image data.
So we had a few projects where basically we looked at comparison between the deep learning algorithms for prediction purposes and compare them with our standard kind of linear mixed model kind of standard prediction algorithms. And in most cases, prediction accuracy was more or less either similar, so we didn't find very big increase or improvements in using those algorithms on genomic data, which is typically in the form of single nucleotide polymorphism markers and these phenotypes, like I show in a few slides. So yeah, we definitely were doing that, and I think in this case it was neural networks themselves that were used in this small master project that we had here. Yeah.
Sorry, I muted myself. Thanks very much. And Tanya and Rachel, do you use anything like that? There is a second question that is asking around convolutional neural networking in particular for detecting image object, to detect image object entity detection.
I must admit the AI side is very, very new to me in terms of image, like trying to find things in images, especially considering I analyze a lot of like underwater footage and stuff. So any ways of doing that would be useful. The only experience that I've had is by using Root Painter, which is a medical AI thing that's used to find stuff in veins or stuff like that, and seeing if we could have used that. But we haven't done any work in that at the moment. And I think my colleague who's done the seagrass research hasn't looked at any AI analysis yet. It's kind of a very new thought as to could it actually detect differences, especially considering the change in like the way the photos would be taken with the different conditions, whether you've got bright sunshine or cloud, and whether that shading would make any difference as to whether it could actually identify it.
Lovely, thanks so much. There was, I think this is in response to Tanya and Rachel's talk, is that you mentioned that people are looking for baseline data that's continuously updated over time. How easy is that to do, and how much effort is required bringing in the new data on top of existing data?
So like for the benthic habitats, for me with rerunning my model, we're aiming to do it annually or however often the data comes in. And now that the main focus has been done, that all the current data is up to that standard, slotting in a new lot of data that's already in the same format at Medin standards has made it a lot easier. Same with like cetacean observations, they're now in the same format. I don't know if Rachel has any other inputs on other data types integrating, updating baseline data.
Yeah, I think I guess there's two sides. There's the integrating new data to create a more complex picture, because the marine environment is so data sparse, it's often data gaps. And then there's the challenge of where there's contradictory data, and it's hard to know whether the original data was spatially inaccurate. So we do a lot of survey work. When we do survey work, we try to survey large areas to try and identify whether that's an issue. And then I guess just collecting data that tracks change over time. So changing fish patterns or like for instance through our stock assessment work, or fisheries use changes over time, and that change is useful to record. But temporal changes generally, whether that's something in mobile things like fishing or cetacean data, it's a challenge to decide what temporal range to use, whether or not we're seeing changes in use or changes in accuracy of the data, and whether we should be actively recording and understanding that temporal change, or whether we should be providing data, the most up-to-date data available.
Lovely, thank you so much. So one question that, or we've got kind of our last couple of questions now to get through. One of them being that, and I always love these kinds of questions, what new tools, existing or novel, would make the biggest impact for you in your research? So what is the one thing tool-wise that would really just make a difference to what you're trying to do right now? Ivan and Bruna, I'll pass over to you first if you can think of anything that might revolutionize.
have to say digital phenotype. So high throughput phenotyping. So for this context that I'm specifically working, like data collection is essential, especially phenotypic information. So the use of AI would really make total difference, I think, for also starting like including additional variables. So yeah.
Brilliant. And Tanya and Rachel?
I think it's just going to come back to that finding ways of analysing the video footage, like not like seagrass is nice, it doesn't move, but different ways of looking at like cetaceans and stuff like that. And we've already seen a massive, just by using drones to analyse, when we see porpoises, suddenly seeing the group dynamics is way bigger than just what you see at the top is one thing, but suddenly having AI look through the footage instead of us would take a lot of strain off in that time capacity when someone's asking about it. It's the rewatching of footage. So when we biocode and identify seabed habitat, each bit of footage is watched 3 times. So if that's an hour worth of footage, that's 3 hours worth of time to analyse. And that's not including the quality assurance side to make sure that what I've said it isn't what I've said it is. So yeah, I think finding ways to automate video analysis and photo analysis would be really useful.
Lovely, thank you. So for our data scientists and AI experts in the audience, maybe there's something there for your thinking hats around that. I did mention I'm conscious that we're running out of time very quickly. It's been a very quick hour with you all. But I just wanted to mention again the Innovation Seed Fund. So this is a jointly funded seed funding from the Data Lab and from SULSA. It's really to support establishing industry collaboration for academics. The lead applicant must be from a SULSA member university, and you can find information on our website about that. I'll pop in a link to the chat as well. But just to let you know, projects is about £5,000 to £15,000 available per project. We're going to fund 2 or 3 projects, and it's really to help with that industry academia collaboration and trying to bring data and life sciences together as well. So I will pop that in the chat. It opens on the 5th of May, so you still have a little bit of time before it opens, and then it will be open until the end of June. So plenty of time still to have a little think about that. But I think it kind of announcing this leads on quite nicely to our final question for today. And I think both of our speakers have been really interesting in showing the applications between data and life sciences and how those applications have real world impact. So whether in Ivan and Bruna's case, that's trying to influence what products get put on market and really working with industry and trying to have that application in the industrial aspect. And then I think with Tanya and Rachel really looking at that influencing policy and affecting policy to make sure that we look after these benthic communities. But our final question was, what do you think could be done to improve industry research collaborations in the future? What are some of the blockers that made it difficult to collaborate effectively? So maybe just I know that there's probably hundreds of things in that we can all be doing better, but maybe if you can highlight 1 or 2 things that are either done well or that are a particular blocker in that sort of collaborative piece. And I guess for Tanya and Rachel to extend that out beyond just industry, but also then in terms of the government and NGO organizations that you work with as well. So maybe I'll start with yourselves, Tanya and Rachel.
I'm going to hand that over to Rachel because she's got way more experience than me with it and probably a lot more deeper insights.
I guess funding is a key blocker, as is data sharing agreements. So we've worked hard to build relationships with industry to enable collaboration. But research is also only ever done over 3 to 5 years at most timescales, which can be not in line with industry needs, and that can be a barrier to collaborate effectively.
Lovely. And any additional thoughts on that, Ivan and Bruna?
No, I actually think it was spot on. So definitely the data component, how to share it, especially when you have more than 2 sides that are involved in the project itself. So kind of data sharing and IP obviously are kind of major components of it. Yeah.
Yeah, absolutely. Well, we have just hit the 12 o'clock mark. So I'd like to thank our speakers again. They were really, really interesting, and I think a great insight into the power of bringing data and life sciences together across sectors. And thank you all for joining us today. Please do keep an eye out on the Data Lab community website and on the SULSA website for any updates. And please do check out the funding call as well. All right, take care, everyone. Thank you.
Thank you.
Thank
Bye-bye.
you.
Thank you.